Okay, so we are going to be talking about chapter 14, which is covering the blood. So the blood is a type of connective tissue. It is suspended in a liquid matrix. So if you take other connective tissues, for example, bone, you're going to have the cells, the bone cells, surrounded by calcium, different connective tissue fibers, okay, which is its extracellular matrix. So blood does count as a connective tissue due to this liquid matrix. Blood is also essential to transport vital substances. It's going to maintain the stability of interstitial fluid. So if you don't remember what interstitial fluid is, that is the fluid surrounding cells within the tissue. So extracellular fluid within the tissues is interstitial fluid. Blood also distributes heat, which we can tell by the fact that when your hands are cold, if you put them in your armpits or your groin, you can feel them warm up. Okay, and that's because there's major blood vessels there that carry a lot of blood. And the amount of blood is going to vary based on a person's body size, their changes in fluid concentration, changes in electrolyte concentrations, and the amount of adipose tissue. Um, all of those are going to affect just how much blood a person has. Blood does make up about 8% of our body weight. For adults, that's about 4 to 5 liters in a female and 5 to 6 liters in a male. As far as blood cells, these are going to form in the red bone marrow and are called formed elements. And they consist of red blood cells or RBCs, white blood cells or WBCs, and then platelets. Even though they're cell fragments, they are considered to be a type of cell. And the other term for platelets that we'll see in a little bit is thrombocytes. Okay, so characteristics of blood. We can see a phlebotomist drawing some blood, and then in the upper right picture, they put the blood tube into a centrifuge. It spins it down so that the most dense or heaviest aspects are going to end up at the bottom and the lightest at the top. So we can see the bottom of the centrifuge blood sample is going to be red blood cells. Above that is the buffy coat, which is white blood cells and platelets. And then at the very top is going to be the plasma. And then on the bottom left, we can see a blood smear. So you can see the red blood cells, the white blood cells, and the platelets. So in a centrifuged blood sample, 55% of the blood is going to be plasma, 45% is going to be red blood cells, and then white blood cells and platelets make up less than 1%. And again, that's your buffy coat. The percentage of red blood cells is called the hematocrit or packed cell volume. Packed cell volume isn't really used as a term very much anymore. The primary one is going to be hematocrit. So you'll a lot of times hear people in the medical field talking about hemoglobin and hematocrit. Okay, so we'll talk about hemoglobin in a little bit, and then the hematocrit is the percent of red blood cells in the sample, which should be 45%. Okay, so the composition of blood. We talked about how it's made up of formed elements and plasma. So the formed elements being 45% of the blood volume is going to be made up of red blood cells, which makes up 95% of that 45. Platelets, which makes up about 4.8%, and then white blood cells making up 0.1%. The white blood cells can further be divided into the 5 different types, which are going to be neutrophils, eosinophils, and basophils, which are your granulocytes, and then monocytes and lymphocytes, which are agranulocytes. As far as the plasma, that is broken up into water being the most volume, 92%, electrolytes, proteins making up 7%. Um, those proteins are albumins, globulins, and fibrinogen. And then we also have wastes, nutrients, vitamins, hormones, gases, and the 3 prevalent gases in the blood are going to be nitrogen, oxygen, and CO2. Okay, so universal precautions. You know, with the coronavirus, everybody's been talking about personal protective equipment or universal precautions. This is, using your PPE, your gloves, your masks, your gown, the use of fume hoods, sharps containers, but it also includes things like safe work practices and hand washing, and that's going to help prevent the spread of infectious disease, whether it's the coronavirus, HIV, hepatitis, um, you know, whatever the case might be. Okay, so here we have the lineage of blood cells. So all blood cells are going to come from what we call the hematopoietic stem cell. Okay, that cell is going to be able to make any type of blood cell. However, in order to do that, it needs to specialize so it can either A, become a myeloid stem cell, or B, become a lymphoid stem cell. So myeloid stem cells are going to be able to create erythrocytes, which are your red blood cells, thrombocytes, which are your platelets, your eosinophils, basophils, and neutrophils, which are three of your white blood cells. It can also create monocytes, and then when monocytes leave the blood and enter the tissue, we know them as macrophages. If it becomes a lymphoid stem cell, um, it's going to either become a T cell or B cell, and B cells that are secreting antibodies are plasma cells. So the cells that are underlined in red are the ones you're going to need to know for your exam. So as we said, the origin of blood cells, they all are going to come from the initial hematopoietic stem cell, and the process of creating new blood cells is known as hematopoiesis. The hematopoietic stem cells, like we said, can give rise to either a lymphoid stem cell or a myeloid stem cell, but they can also copy themselves to create more of these hematopoietic stem cells. So they have three choices. A, copy themselves, create more hematopoietic stem cells. B, become a lymphoid stem cell, which will then eventually give rise to either a T cell or a B cell, or become a myeloid stem cell and become either a red blood cell, a platelet, or 4 of the 5 white blood cells. Okay, so red blood cells, these are known as erythrocytes. Their shape is a biconcave disc. So if you notice the red blood cell in the top right, it looks like a donut with the center hasn't been punched out fully. Okay, that's what it means by biconcave disc. And a third of the red blood cell is made up of hemoglobin. If that hemoglobin has an oxygen attached, it's known as oxyhemoglobin. If it doesn't have an oxygen attached, it's known as deoxyhemoglobin. Red blood cells lack a nuclei and mitochondria, meaning they can't divide, they can't produce large amounts of energy, so whatever energy in the form of ATP that they do need, they create through glycolysis or the splitting of glucose. Normal red blood cell counts. So whenever we talk about red blood cell, white blood cell, or platelet counts, it's always going to be the number of cells in a microliter of blood. A microliter of blood is about the amount that would be on the end of a needle if you had just drawn up some meds and just had that tiny little bubble at the very tip of the needle. That's about how big a microliter is. So in that microliter, males would have about 4.7 to 6.1 million red blood cells. Females have about 4.2 to 5.4 million red blood cells, and children would have about 4.5 to 5.1 million red blood cells. As far as your test goes, I would remember 4.5 to 6 million as being the normal red blood cell count. Now, these red blood cell counts are useful in the diagnosis of disease and how that disease might be progressing. If the number is too low, or too high, this is going to give us a good idea of, what kind of condition the patient has, or is it getting better, is it getting worse? And additionally, the number of red blood cells is going to tell us about the blood's oxygen carrying capacity. So if the person's organs aren't getting enough oxygen and they're breathing just fine, it may be due to the fact that there's not enough red blood cells to carry that oxygen to the organ in need. Okay, so erythropoiesis, again, this is the process of creating blood cells. So hematopoiesis was blood cells in general. Erythropoiesis is red blood cells specifically. And this process is started by the release of the hormone erythropoietin. Okay, so this is going to be released when there's not enough red blood cells or not enough oxygen in the blood. The kidney is going to release it saying, hey, we need some more red blood cells. The hormone travels to the red bone marrow and tells the red bone marrow to start producing more red blood cells. And this process takes a couple days between when the signal is first initiated and the release of erythropoietin to when the red blood cells are actually produced and ready to go. Okay, so dietary factors affecting red blood cell production. You need vitamin B12 and folic acid. Without these, you're going to get different types of anemias due to the fact that the cells can't grow and divide. And then iron is required for the synthesis of hemoglobin. So without iron, you can't create hemoglobin. Okay, so different types of anemia. The first one we're going to talk about is on the far right of the slide, the sickle cell disease. If you notice by the arrow, there's an oblong red cell. That is a sickled red blood cell, and this is going to happen in times when the patient is sick, when they are not breathing best, and that's going to cause sickling of the red blood cell. This is typically found in sub-Saharan Africa. Races that live in sub-Saharan Africa are used to, as well as around the Mediterranean Sea. So people from Italy, Northern Africa also can have sickle cell disease. The reason for that trait is it makes the person immune to malaria. So malaria, which is carried by mosquitoes and is in your hot tropical regions, it cannot reproduce inside the red blood cell like it normally would if the red blood cell is sickled. In the middle, we have iron deficiency anemia, and if you notice, we have a large white spot in the middle of the cells, and that's because there's not enough hemoglobin in that cell to actually give it coloration throughout the entire cell. So if you remember the biconcave disc, the center is thinner. Well, in this case, there's not enough hemoglobin in that thin center to actually give it color, so the light passes right through. So iron deficiency anemia obviously is due to a lack of iron. The other one you need to know is pernicious anemia. This is due to a lack of vitamin B12, and it's going to cause large, fragile red blood cells. Okay, so events in the destruction of red blood cells. So what you need to know from this chart, it's a little bit, confusing, is that after about 120 days, red blood cells are going to start to get damaged and die. And when they die, the hemoglobin needs to be broken down as well as the cell. So we're going to split the hemoglobin into heme and globin. The globin goes away and can be reused later. The heme is then split even further into iron and biliverdin, which has a greenish color. The iron is going to be stored in the liver to be reused later. And the biliverdin is going to start heading towards the gallbladder because it becomes a bile pigment. Some of that biliverdin is going to be converted into bilirubin, which is why if you've ever been sick and thrown up so much there's no food left, and you throw up a little bit more and you get that greenish or orangish color in your vomit, that's the biliverdin and the bilirubin. They are secreted with your bile and normally are just broken down in the GI tract, and as we'll learn in a couple chapters, that's what gives your feces its lovely brown color, because they have been broken down by bacteria to form a brownish color. So the key here is to remember that hemoglobin is split into heme and globin. Heme is broken down into iron and biliverdin. Biliverdin can be further converted into bilirubin. You do need to know the colors, so biliverdin was green, bilirubin is orange. Okay, so here we just have the life cycle of a red blood cell. So we know that the red blood cell is going to be created in the bone marrow. It's going to be released from the bone marrow. It lasts about 120 days. At the end of that 120 days, The hemoglobin inside is going to be broken down into heme and globin. The heme is further broken down into iron and biliverdin, and some of the biliverdin is converted to bilirubin. Okay, so what you need to notice on this slide is, first of all, the fact that there are 4 subunits for hemoglobin. Okay, so we can see over here on the left we have a beta 2 and a beta 1 chain, an alpha 1 and an alpha 2 chain. You don't need to remember the names. You do need to remember there are 4 subunits in hemoglobin. You also need to know that within each subunit is an iron core. Okay, so without iron, the subunits cannot be made. Okay, and then each subunit can carry 1 oxygen. So hemoglobin as a whole can carry 4 oxygen. You do not need to know the molecular structure of biliverdin and bilirubin. As far as the heme, you just need to remember that the Fe, which is in the middle, is iron. Okay, so as far as the types of white blood cells, we're going to have granulocytes and agranulocytes. Granulocytes have granules in their cytoplasm and typically have a short lifespan. Agranulocytes do not have granules in their cytoplasm. So your granulocytes are going to be neutrophils, eosinophils, and basophils. Agranulocytes are lymphocytes and monocytes. As far as the hormones that cause the creation of white blood cells, you don't need to remember the interleukins and colony stimulating factors. Okay, so neutrophils, big things to know about neutrophils are, number one, they're the first to arrive at the site of an infection. So they very quickly get to the scene of where there's been injury or illness. They're strong phagocytes, meaning that they're going to engulf pathogens, cellular debris, etc. Once they engulf it, they're going to break it down in their different lysosomes, things like that. And they're the most common. So 54 to 62% of all circulating white blood cells are going to be neutrophils. And then lastly, they're elevated in bacterial infections. So if you take someone's blood and they have a high neutrophil count, either A, they have a new infection, something that just came on, which would be why they're high, or they have a bacterial infection. Okay, so eosinophils, these are going to moderate allergic reactions. Moderate means control, so they can make the allergic reaction either better or worse. And they're also going to defend against parasitic worm infections. And then the last granulocyte are our basophils. These release histamine and heparin. Histamine is going to stimulate inflammation, and then heparin is going to stop blood from clotting. So the inflammation is going to try and wall off that area that's been infected or injured, while at the same time bringing in the different cells and molecules needed to help fight off infection and start the healing process. Heparin is going to help keep blood from clotting. We want the blood to continue flowing in that area so that the different white blood cells, antibodies, etc. can get into that area. Okay, monocytes, this is the first agranulocyte. It's going to be a strong phagocyte just like your neutrophils were. They're going to phagocytize bacteria, dead cells, debris. They live for a few weeks to a couple months. And when they leave the bloodstream, they're then known as macrophages. So when they're in the bloodstream, they're called monocytes. When they leave the bloodstream and enter the tissues, they're known as macrophages. So macrophages and neutrophils are going to be your strong phagocytes. And then the last type of white blood cell are lymphocytes. These are agranulocytes again, and they're very important when it comes to your immune system. T cells are going to directly attack pathogens and tumor cells, whereas B cells are going to produce antibodies that go out and find the pathogen and help destroy it. These live for many years, which is why you can be exposed to a condition and the next time you're exposed, you won't become ill. Okay, so functions of the white blood cells. First is diapedesis. So this is just the ability of the cell to move to the edge of the blood vessel and then squeeze out of the blood vessel and move towards the site of infection. Cellular adhesion molecules, these are proteins that form like a breadcrumb trail to an injury site. It's very strongly related with the positive chemotaxis at the bottom. So the cellular adhesion molecules are the breadcrumbs. Positive chemotaxis would be the ability to follow that breadcrumb trail to where the white blood cells need to be. We talked about phagocytosis and how that's when a cell engulfs either pathogens, cellular debris, or um, things like tumor cells and destroys them. And then inflammatory response, we talked about how this is going to be controlled by basophils and their secretion of heparin and histamine. And it's going to have things like swelling due to increased blood supply to that area. The redness is due to that increased blood supply. The pain is due to some of the chemicals that are released at that time. And it also allows for capillary permeability so that the white blood cells, antibodies, different chemical molecules can get through the wall of the capillary and into the affected tissue. Here we just have an image that kind of shows this process. So, the person got a splinter. There are germs or bacteria on the splinter. The bacteria start to multiply in the tissue. Neutrophils very quickly come to the area and start to phagocytize the bacteria to help and try and limit the infection. Okay, so white blood cell counts. Normal white blood cell count is 3,500 to 10,500 cells per microliter of blood. If it's over 10,500, it's known as leukocytosis, and this is typically due to acute infections. Vigorous exercise and loss of body fluids is very rare for causing a high white blood cell count. Leukopenia is when you have a low white blood cell count, less than 3,500, and this could be due to things like typhoid fever, flu, measles, mumps, chickenpox, AIDS, polio, and anemia. Now, when we measure the white blood cells, we're not just measuring the total number, we're also going to figure out what the different percentage of the types of white blood cells there are. So if there's a high neutrophil count, we'll know that it's either something very acute or a bacterial infection. Eosinophils, well, it's allergies or parasitic worm infection. If there's too low a number of helper T cells, it could be due to HIV, and so on. Okay, so abnormal white blood cell numbers. The big ones to remember are elevated eosinophils are going to be your parasitic worm infections and allergic reactions. Elevated neutrophils being bacterial infections. And if someone has too few helper T cells, it's typically related to HIV and AIDS. Okay, so two types of leukemia. Leukemia affects the white blood cells. You can have lymphoid leukemia or myeloid leukemia. Lymphoid leukemia is going to affect the lymphocytes. There's usually too many of them, and as a result, they're going to crowd out the ability of the myeloid stem cells to create things like red blood cells, platelets, and the other four types of white blood cells. On the other hand, myeloid leukemia is going to do the opposite. It's going to make it so that the bone marrow cannot create enough of the lymphocytes or your T cells and B cells. Treatments, chemotherapy, now they're getting different drugs that can target the enzymes that are specific to cancer cells. And then the gold standard is bone marrow or stem cell transplants. These pretty much cure the condition. However, it can be tough to find someone who is a match donor that can donate the bone marrow or stem cells unless you have a twin or very closely related family member with the same genetic characteristics. Okay, so platelets. Platelets are fragments of the megakaryocytes. They're going to be produced in response to thrombopoietin. Okay, don't need to worry about the hemocytoblast to megakaryocyte to thrombocyte. You just need to remember megakaryocytes break apart to form thrombocytes. They lack a nucleus and are very small, even smaller than red blood cells, and the normal amount is about 150,000 to 350,000 platelets per microliter of blood. Their function is to help in hemostasis, which is the stoppage of bleeding, by sticking to broken surfaces and forming a plug. And they also release serotonin, which is going to cause the smooth muscle in the walls of the broken blood vessels to contract. Okay, take a look at table 14.5 and study it for your exam. Now plasma, this is a clear straw-colored liquid. It's the liquid portion of the blood. It makes up 55% of your blood volume, and it's 92% water. It also contains organic and inorganic chemicals. So organic chemicals are those containing carbon. Inorganic chemicals are those that do not contain carbon. It's also going to transport nutrients, gases, hormones, and vitamins, and it helps regulate fluid and electrolyte balances, as well as maintain your body's pH. Okay, so plasma proteins, first of all, they're not typically used as an energy source. So if for some reason you're in the middle of the desert and you're starving, your body will start to break down your skeletal proteins, so your muscles. However, the body doesn't typically use the proteins in your blood because it becomes very, very, very bad for the body when it does. So the proteins are albumins. Okay, these are going to help keep the liquid portion of the blood inside the blood vessels. That's what it means by help maintain colloid osmotic pressure. You then have globulins. The one that we're going to pay attention to mainly is the gamma globulins. Those are going to be your antibodies for immunity produced by your B cells. And then fibrinogen is produced by the liver, and we're going to talk about that as it has a role in blood coagulation. Okay, so gases and nutrients. Most important blood gases are oxygen and carbon dioxide. We do have nitrogen in our blood, but it does not have any effect on the body, and the body cannot use it, so it's considered to be inert. Plasma nutrients, so amino acids, these are used to build proteins. Simple sugars are used to build carbohydrates. Nucleotides are used to build DNA, RNA, genetic material. And then lipids, we have our fats or triglycerides, the phospholipids that make up your cell membrane, and cholesterol, which has a variety of functions in the body. Non-protein nitrogenous substances, so these are molecules that contain nitrogen but are not proteins. So urea is a byproduct of protein breakdown and accounts for over half of the non-protein nitrogenous substances. It's found in high levels in the urine. Uric acid is a product of nucleic acid catabolism, meaning it's from the breakdown of genetic material. High levels of uric acid are related to gout. Amino acids, these are the product of protein digestion, so these are the building blocks for the proteins. Creatine is going to store energy in its high energy phosphate bonds. It can be used to regenerate ATP in the muscles. And then creatinine is the product of creatine breakdown. BUN, do not call it BUN, it's BUN, blood urea nitrogen, is used to indicate whether the kidney is healthy or not. So typically if we're trying to figure out how healthy someone's kidneys are, we'll use creatinine and BUN. If they're high, it typically means that the kidney is not working very well. Okay, so plasma electrolytes, remember that electrolytes are called that because of the fact that when they ionize in water, they can conduct electricity. So if you put enough salt into a pot of water. You could actually take plugs, the ends of plugs, put it in, and it'll conduct electricity and complete the circuit. Do not do that at home though, please. They are going to be absorbed from the intestine or released as byproducts of cellular metabolism. So the common ones are sodium, potassium, calcium, magnesium, chloride, bicarbonate, phosphate, and sulfate. Sodium and chloride are the most abundant, and that's typically because of the fact that your table salt is a combination of sodium and chloride, so we eat large amounts of that, and thus we have high levels of both of them. Okay, so hemostasis is the stoppage of bleeding. There's 3 different steps to hemostasis. The first is avascular spasms, so the blood vessel spasms, making the lumen more narrow and blood loss decreases. Then comes the formation of a platelet plug, and then lastly is blood coagulation. These are effective in small blood vessel injuries, not if you have major injury to your arteries in your neck or your chest. It won't work well enough for those. Okay, so vascular spasm, that's going to be initiated or stimulated by the cutting or breaking of the blood vessel. The smooth muscle in the wall of the blood vessel is very quickly going to contract. This slows down blood loss, and if it's a small enough blood vessel, it can close it completely. Response is going to last a few minutes, but it can last up to 30 minutes, and this allows time for the platelet plug to form. Serotonin from the platelets is also going to help with the vasoconstriction to reduce blood loss. The second step is the formation of a platelet plug. So when the platelets are exposed to the collagen fibers in the wall of the blood vessel, they're going to become sticky. So they start to stick to the walls, stick to each other. And as you can see, eventually enough of them stick together that it actually causes a plug and blood cannot get past this. This will last until hemostasis has actually, or not hemostasis, blood coagulation has occurred, and then can be broken down. And the last step is blood coagulation. So this takes about 5 to 15 minutes to occur. It forms a blood clot in a series of reactions, with each one stimulating the activation of the next, which is why we call it a cascade. There are 2 different start points. There is an extrinsic and an intrinsic mechanism. Okay, so extrinsic comes from outside of the blood vessel. Intrinsic starts on the inside of the blood vessel. The chemicals used in the coagulation cascade are called clotting factors, and most of them require vitamin K. One medication that works on this area is warfarin and Coumadin. They're actually a faulty version of vitamin K. When the body builds it into the clotting factors, they aren't effective, and so it makes it so that the person cannot form clots, which is good because that can help prevent strokes or heart attacks in people who have other underlying conditions. The major event in blood coagulation is the conversion of soluble fibrinogen into insoluble threads of fibrin that trap the blood cells, as you can see in the picture below. So the yellow threads are the fibrin, and then they have caught the red blood cells in essentially a spider web. Okay, so table 14.7 goes through the different hemostatic mechanisms. Okay, so extrinsic clotting mechanism, again, this comes from the outside of the blood vessel, typically due to trauma. You don't need to remember tissue thromboplastin factor 3. The cascade's going to begin, and then it gets to a point that we call the common pathway, where thrombin is going to convert fibrinogen into fibrin. Okay, the fibrin is going to trap all of the different blood cells and platelets, forming a blood clot, and this is going to stay there until the tissue has sufficiently healed. This is an example of positive feedback. So the more you start to clot, the more your body wants to clot. and that's not good. So in a few slides, we'll talk about ways that the body actually prevents the clotting from uh, starting like a runaway process. Intrinsic clotting mechanism. So this can start without tissue damage. It's typically due to contact with a foreign surface inside of the blood vessels. So an atherosclerotic plaque, some type of um, excess cholesterol buildup. It could be an embolism, a clot that broke off from somewhere else. But the key point is it starts inside of the blood vessel. You don't need to remember that it's triggered by Hageman factor 12. Just like the extrinsic clotting mechanism, it's going to start a sequence of reactions occurring, and it has the same result as the extrinsic clotting mechanism, which is thrombin converting fibrinogen into fibrin. Okay, so table 14.8, blood coagulation. You need to know the trigger. Okay, so extrinsic was damage to the blood vessel, and then intrinsic is the contact with a foreign surface. You do not need to know row number 2. You do not need to know row number 3. You do need to know row number 4, which is the formation of thrombin. And thrombin is then going to convert the fibrinogen to fibrin. So both mechanisms have the same result, and that's the conversion of fibrinogen to fibrin to form the clot. Okay, so here's the huge long actual clotting mechanism with all the factors. Okay, again, you just need to remember extrinsic and intrinsic. Remember, extrinsics from outside of the blood vessel, intrinsics from the inside. And then you need to know that thrombin is going to convert fibrinogen to fibrin. Okay, so those are the important aspects to know for the blood clotting cascade. You guys do not need to know all of the different clotting factors, so you can just cross that out. Okay, so the fate of blood clots. As the blood clot forms, it's going to pull the edges of the blood vessel back together, okay? And then it's going to start to heal from there. Once the clot has outlived its usefulness, plasmin is going to digest the fibrin threads and dissolve the blood clots. It's like a chainsaw that you take to the blood clot. But a thrombus is an abnormal blood clot that forms in a blood vessel. So this is a clot that formed, it did not have a purpose, and it should not be there. If that thrombus was to break off and start moving throughout the blood supply, it would be known as an embolus. Okay, so abnormal blood clot formation, so thrombosis. This is a blood clot where it does not belong. It does not need to be supplying a vital organ, the brain or the heart. Deep vein thrombosis is when you get a clot in your calf muscle area, and that is not supplying your brain or your heart. Infarction is death of the tissues around a blood vessel that's been blocked. So, the actual name for a heart attack is a myocardial infarction. So in this case, the myocardial tissue does not have sufficient blood supply, oxygen, nutrients, and the tissue starts to die, which is why we call it a myocardial infarction. An embolism, it can be a blood clot that travels and then blocks a blood vessel in an organ, away from where it was created. So the best example is a pulmonary embolism. So if someone has a DVT, deep vein thrombosis, if it breaks off, the first area it typically tends to get stuck in is the lungs. And then atherosclerosis is the buildup of cholesterol and fat between the inner and middle lining of a blood vessel and can help with the formation of a thrombus. Okay, so deep vein thrombosis. This is a clot formation due to pooling of the blood, typically because the blood has become stagnant. So if you took a long trip or you're bedbound, you're in the hospital, a nursing home, you can form a deep vein thrombosis. If it breaks off, it can form a pulmonary embolism, where it blocks part of the blood supply to the lungs. It can actually cause death. Symptoms of a DVT are going to be things like deep muscle pain, cramping, redness, swelling, and dilation of the surface veins. The clot can break off days or hours after formation. Okay, so prevention of coagulation. The smooth lining of the blood vessels is one of the best ways that we prevent having any accumulation of platelets and clotting factors. If a clot does form, the fibrin adsorbs, essentially means it latches onto the thrombin and builds it right into the clot, so it can't go anywhere else and start another clot forming. And then any thrombin that is left over can be inactivated by what we call antithrombin. And that's just going to latch onto thrombin and prevent it from binding to fibrinogen. Lastly, some of the cells, like our basophils, secrete heparin, and heparin's an anticoagulant that prevents the formation of a blood clot. It can't break down a blood clot, but it can prevent it from forming. Okay, so those factors we just talked about are in table 14.10. Okay, so blood groups and transfusions. So blood grouping and transfusion can be kind of difficult to understand, so I'm going to post some different images, videos, things like that that might help you understand the blood groups and their transfusions. So in 1910, we identified the ABO blood genes. So these are carbohydrates or markers on your red blood cells that determine your blood type. Prior to that, we did not understand that there was different types of blood. We just thought blood was blood. There were some people who, thought that your blood differed based on your races. So for a very long time, African American blood was not accepted, even though they had determined, back in 1910 that the blood was the same. It just depended on the different antigens on your red blood cells. Okay, so an antigen is any molecule that can create an immune response. So all of our red blood cells have antigens on it. Well, most of them do. We'll see in a few minutes. And that can actually cause an immune response if it's given to somebody with a different blood type. Antibodies are proteins created by your B cells that are found in your plasma and are going to attack the red blood cells that are given to you from somebody else if it's not the right blood type. Okay, when it attacks the red blood cells, the combination of the antibodies and the red blood cells causes agglutination, which is a clumping of the red blood cells. This is really bad for the body and can cause a lot of health issues. There are 33 known antigens on red blood cells. However, the ones that we really only pay attention to are ABO and RH, and that's because if you give the wrong ABO or RH group to someone, it can cause really bad adverse effects, including death. The other antigens really don't cause much of a reaction, so we don't really worry about them. Okay, so ABO blood groups, these are defined based on the antigens on your red blood cells. So we have an A antigen and a B antigen. They're carbohydrates, meaning they're sugar chains, okay? And your antigen is going to determine your blood type, okay? So if you have the A antigen only, you're going to have blood type A. If you have only the B antigen, you're going to have blood type B. If you have both A and B antigen, you're going to have blood type AB. And if you have no antigen, neither A or B, your blood type is O. Okay? You always have to have something that says A and something that says B. So if you have only the A antigen, you have to have the anti-B antibody. If you have only the B antigen, you have to have the anti-A antibody. If you have both A and B antigens, well, you're not going to have either anti-A or anti-B. And if you have no antigen, you're going to have both anti-A and anti-B antibodies. Okay, so something that's A, something that's B. It could be both A and B are antigens, or it could be both A and B are antibodies, but you have to have at least one of each. The other part to remember is that the antigen is always going to be in your blood type's name. So if you have A antigen, your blood type's A. If you have B antigen, your blood type's B. If you have A and B antigen, your blood type's AB. O doesn't have either antigen, and that's why it's not in the name. Okay, so the most common blood types in the US, this is just for your own personal satisfaction, and it's typically going to be O. Okay, so here we can see the 4 different blood types. We have type A, B, AB, and O. So we can see that in type A blood, we have the A antigen on the surface of the red blood cell, and then we have these anti-B antibodies floating around in the plasma, just there in case somebody accidentally gives you type B blood. Same idea with type B blood. The B antigen is on the surface of the red blood cell, and then we have the anti-A antibody floating around in the plasma, waiting in case the person's given type A blood accidentally. If we drop down to the bottom left, we can see type AB blood. So because this person has both antigen A and antigen B, they don't have either antibody. If they did, it would cause major agglutination and possibly death for that patient. So people with type AB blood have both antigens on the red blood cell, but no antibody. And then lastly is type O blood. So these people don't have either A or B antigen on the red blood cell. So they have both the anti-A and the anti-B antibody floating around in their plasma. Okay, so agglutination. We can see at the top that they decided, hey, we're going to put type A blood into a patient who has type B blood. So now all these anti-A antibodies are going to attach to the red blood cells. And on the bottom left, you can see what a normal blood smear of our blood looks like. And then on the bottom right, it shows you what happens when you give someone the wrong blood type. This is what agglutination looks like. So instead of the blood cells being all nicely spaced out and doing their thing, they're all clumped up and aren't working well. Okay, so compatible blood types for transfusions. We're not going to go into adding the RH to the ABO system right now. We'll talk about it in general, but as far as your exams go, you don't need to remember, A positive, A negative, B positive, B negative, etc. Just A, B, AB, and O. So if you have type A blood, we would prefer to give you type A blood, but you could also get type O in an emergency. If you have type B blood, again, preferred would be to give you B, but you can also receive O in an emergency. Type AB blood, again, we prefer to give you type AB. However, if needed, we could give you A, B, or O. And then lastly, for type O blood, preferred is to give you type O, and there's no alternates. Okay, so patients who have type O blood can only get type O blood. So type O is our universal donor, meaning that it can be given to anyone. Okay, and that's because of the fact it has no antigens on its surface. So there's nothing for the antibodies in a person's plasma to react with. Type AB is the universal recipient, meaning that they can get any type of blood, A, B, or O, and that's because of the fact that they have no antibodies in their plasma to react with any of the other blood types. Okay, so the Rh blood group, this is completely and totally separate from ABO. They have no relation to each other. However, we do put them together for ease of communicating information. So the Rh blood group is what's going to give you the positive or negative on your blood type. Okay, so if you have the Rh antigen on your red blood cell, you would be positive. If you do not have the Rh antigen on your red blood cell, you'd be negative. So if you're type A positive blood, it means you have the A antigen on your red blood cell and the Rh antigen on your red blood cell. If you're A negative, you would have the A antigen on your red blood cell, but no Rh antigen on your red blood cell. Okay. Rh really becomes a problem, or it used to be at least a problem, when mothers who are Rh negative had Rh positive kids, and that condition is known as erythroblastosis fetalis. So in erythroblastosis fetalis, we have a mom who's Rh negative, dad who's Rh positive. This causes the ability for the mom to have an Rh positive child. When the child's being delivered, some of the blood between mom and the child is mixed, and mom starts to develop antibodies against Rh. Okay. The next time she is pregnant with an Rh positive child, the antibodies are going to cross the placenta and start to attack the child, causing birth defects or a spontaneous abortion. Okay. This can only happen if mom is Rh negative, dad is Rh positive, and it only happens to the second Rh positive child on. So if mom's first baby is Rh positive, mom's going to get sensitized. If she only has Rh negative kids after that, none of her kids will ever be affected. However, if she does have an Rh positive child later, that child could be attacked. We have developed a medication to prevent this known as Rogam. So Rogam is an injection of antibodies that are actually going to attack and destroy mom's antibodies. Okay, in that case, it's preventing her from having any antibodies to cross the placenta to attack the child, and the child comes out just fine. Okay, so as far as inherited blood disorders, the only one you really need to know about besides sickle cell that we talked about before is hemophilia, and this is when the patient is missing one or more of their clotting factors. This is typically only going to affect men. Most of the clotting factors are found on the X chromosome. Men only have one X chromosome, females have two. So females typically will have a good set of genes for the clotting factors on their second X, whereas men, because they only have one copy, typically will have the signs and symptoms of having hemophilia. This used to be a huge issue because the bleeding that's caused could lead to death. You have cases back in the medieval days. A lot of the royal families had hemophilia due to the amount of inbreeding, trying to keep their bloodlines pure, where a child would be injured and they would bleed and bleed and bleed for days, and then finally days later they would die because they had lost too much blood or couldn't fight off, the symptoms of shock. So hemophilia is the inability to clot normally. And then sickle cell, again, we said this is when the red blood cells change shape based on being sick or having a low oxygen situation, and it can cause anemia, pain, and other symptoms. Okay, so that's everything. Please make sure you message me with any questions you might have. Watch the other resources I'll be putting up this week, and come to the classes with any questions, because we won't be doing the entire lecture in class. It'll be question and answer based. Okay, thanks.